everybody. Thank you so much. Hi, Catherine Lee. I recognize that name. All right, I'll get started here. So what I want to do and what I do at the beginning of each talk is kind of set expectations. If you're thinking of giving a talk, do this. Tell the audience what you're going to tell them, and then tell them again. Anyway, so I'm going to tell you in two parts here. So first part, I'm going to tell you a little bit about who I am and how I got here, and what I do as the academic affairs director at Fox Chase, particularly the skills needed, what I like about my job, what I hate about my job, and then maybe some advice of how to get there. And since the audience is mostly high school students, I wanted to tell you about a thing that I'm really passionate about, one of my favorite programs, and that's called TRIPS, for Teen Research Internship Program. And also there's a couple people on the line that are currently involved. Again, hi, Catherine. That was the only name I saw, but then I stopped looking at the chat. But I'm going to tell you about what TRIP is, who can apply, how you apply, and what we actually do there with fruit flies. And it's basically hands-on research opportunities for high school students. Okay, so this is me. I just putting this up so that you can get my email address if you want to get in touch with me after this. Also, if you're on social media like Twitter or LinkedIn, there are my handles. On Instagram, I'm just Trip Initiative because I don't like having my own thing, but feel free to reach out and connect with me. I will also put this up at the end. Okay, so I always think it's important to start a talk just to give context of tell you what things drive me. So I'm really passionate about communication. So being able to connect with people, particularly about scientists, because I feel like there's a gap between what scientists think and what the community thinks. So opening up that dialogue in between people. And how I get to that is really teaching researchers, scientists, medical professionals at the Cancer Center to be better communicators, and then also being involved in projects or programs to get people involved, get stuff in their hands, and talk about science. And the reason I'm really passionate about these things is because these types of images. So I feel like how we teach science right now is boring. It's a bunch of facts, memorization, and it's not a lot of application. It's not a lot of idea generation. And candidly, it's not a lot of fun. And if you've been listening to the news in the last, few years, haven't been living under a rock necessarily, you see that there's a lot of people that discount the science that's out there, particularly when it comes to climate change. And I think a lot at the heart of this is about how information is delivered and who is delivering it. So I want to make sure that the scientists of tomorrow can actually speak good, be engaging, and then listen and have their ears open to what the community needs. I'm also interested in retention of students in STEM, so science, technology, engineering, and math. And a thing that runs through all of my programs is a real mandate for diversifying, particularly the upper echelons of STEM, because right now they're just a bunch of white dudes. Okay, so how I define myself is I'm a research scientist first. That's all of my training. I will tell you, though, the job I do right now is not what my training is in. So just put that in the back of your brain for a little bit. So as Dave said, I have a bachelor's degree in genetics and cell biology. So I'm really interested in how cells talk to each other, how molecules on cells interact, and what it does to the cell. Anything about cells, usually it's my jam. I have a PhD in cell biology, particularly studying the cell cycle. So how cells divide, make more of themselves. And after you get a PhD, just like after you get an MD, you're not done. You got more training to do. On the PhD side, it's called a postdoc, so postdoctorate degree. And mine is focused on immunology. And I actually studied this reaction that's shown here on the right-hand side. Hopefully you guys can see my little mousey moving around. That NK cells are natural killer cells. They rove around your body and look for either your cells that have become transformed or cancerous or infected with certain types of virus. And then what they do when they find those cells is they glom on, like this NK cell, onto a tumor cell, give it a big hug, and then whisper to the cancer cell, kill your cells. Hopefully you heard that. And the tumor cell actually goes through cell death. So that interaction, particularly under a microscope, is fascinating. So that's what my postdoctoral training was in. But you can see still about at the cellular level. So as I said, what I actually do for a living is not just what I was trained to do. So again, I'm a research scientist. So these are the types of things I do when I'm being all sciencey. So I plan experiments, I actually do the experiments, I talk about the results, I read the literature, and I do lots and lots of writing. But I'm also an educator. So I teach classes. So I'm actually teaching a class at a Chestnut Hill College right now on clinical trials. But I create programming and I interact with students all the way from high school all the way to postgraduate. So people on the medical and on the research tracks. And I also talk to the community about Fox Chase. That's FCCC here is Fox Chase Cancer Center where I work. And I talk about science and also talk about the role of the Cancer Center in the community. But the last thing I am, and this is the thing that I have no training in, which is fun, is that I'm a director or administrator. So I build programs, I manage people, I mentor people. I have to manage budgets. Oh man, that's tough sometimes. I think about events, I create new partnerships, I fundraise, I cultivate donors, and basically I tackle any problem that has kind of been put in front of me. And I do credit my ability to be the director of programs to all of the stuff that I struggled through as a research scientist. So I want to say that if you learn nothing else, so when you hear these career talks, you're like, that person has it together. They went from point A to point B and everything worked out, and it's going to be really hard for me to get there. For me, totally not true. So if you learn nothing else about me, is that the secret of my success really comes from my mentors. So I'm going to show you pictures of 4 people. You don't know them, but I'll tell you a little bit about them. So James Mickelson, we always called him Mick, was my research advisor in undergrad. And he was the dude that really actually opened the door and let me into the lab. I started as a dishwasher in his lab. I know, very, very auspicious start. And within 1 week, I was doing some experiments because I was so annoying. Because I was like, what's that? What's that? What are you doing? I still had to do the dishes, just FYI. Tintin was my graduate student mentor. And actually, before I move on to Tintin, I do want to say that Mick was the first one that told me about that there was sort of, after undergrad, there was additional training and that I should consider maybe getting a PhD. I'd never thought about it before. When I went to graduate school, Tintin was my advisor. What I really remember about her is she told me that not all scientists are the same. They don't look alike. We can be different, and that I was sort of outside of the mold and that I should embrace that. And I carry that advice with me because sometimes it's hard not to be like everybody else. Carrie is my postdoc, who is my postdoc mentor. So he taught me all about the language of immunology, which sometimes it really does seem like a completely foreign language. If you've ever been to an immunology talk, you'd understand. Anyway, he also gave me the space to develop lots of other skills. And then Glenn, he is the chief academic officer at Fox Chase, and he's really allowed me the space as well to develop professional skills and just sort of explore and build as I see fit. So I am where I am today because of these 4 people. So I urge you to think about the people in your life who can be mentors, who can be good for you, who you can learn from, and keep them close. Okay, so that's the other thing that I wanted to mention was that your career path is not going to be a straight line. It doesn't go from A to B. So you don't start in school, you do some other stuff, and then you end up in your career path and you're like, woo, I did it. Everything worked out. Nah, it's usually something like this. You take a couple of steps forward, you take a couple steps back, and some decisions that may seem bad at the time, when you're looking back on them later, can actually be really good. So I'm going to tell you a little bit about some of the places that I've lived and the things that have sort of influenced me to the place that I am now. And hopefully you all find this interesting. Otherwise, again, type into the chat box and say, focus on something else. Okay, so here's a map of the United States. Hopefully this seems familiar to you. So I'm from a suburb of Minneapolis called Apple Valley. It's right there. There are a bunch of apple trees there. I don't know if that's the name, that's why. When I was little, I was really interested in medicine and science. I like to figure things out. I like to do experiments in the kitchen at night and then not clean up my messes, my parents tell me. And I'd say I always had an aptitude for science. Like the science classes totally made sense to me. I struggled in history, did not make sense to me, just seemed like a bunch of dates that I wasn't that interested in. And I struggled in math, but I really, really liked science, particularly biology. I got A's and B's in school. I can't say that I tried super hard beyond the classes that interested me. And I was involved in some extracurricular things, but I never took an AP class. I didn't think that was important at the time. I wanted to hang out with my friends and just be. So when I was little, I really thought I'd probably be a scientist, but I wasn't sure exactly what kind. I knew I was interested in people, in cells, in tissues. I certainly wasn't interested in, say, like planets and other galaxies. And I wasn't interested in like individual molecules, so maybe like chemistry. I was more interested in the things that I could see in our bodies and how things worked. So what was really influential for me in high school was that I thought I had it all set in junior year. Like I was going to go to college in some other state that there wasn't as much snow. I was going to be elsewhere. So I decided to move to Ecuador for the summer between my junior and senior year and be kind of an exchange student and check things out. And just if you don't know where Ecuador is, it's in South America. It's right off on the equator, ergo the name. So from this experience, I had never been that far from home. And I saw some things there that really made me reevaluate where I wanted to be. And so I came back and I said, no, I'm not going anywhere. I'm going to stay nearby and I'm going to really focus on maybe research and maybe science. So I ended up going to the University of Minnesota, which was a big powerhouse school, particularly in research. And I majored in genetics and cell biology, kind of a theme here of the things that I told you I was interested in. So just some highlights. The things that I did in undergrad is that I joined a lab almost right away. And again, I started as a dishwasher. You got to start somewhere. And it took me 3.5 years to graduate. And in that time, I worked in the lab all of the time, either half-time, so 20 hours a week or more. And then when I graduated early, I stayed on for an extra 9 months to finish working in the lab and finishing up some papers. And I have to say, the lab, I loved it. I loved almost every minute of it, even the washing of the dishes. It was so much more interesting than being in class. And what was kind of cool is that the stuff that I was learning in class, I could then see in the lab and I could do with these guys, do with my hands. And that really helped the information stick in my brain versus reading it on a page. Dave, I see that the chat box keeps lighting up. Is there anything I need to pivot? You're muted. You're still muted.
Man, a year into this and I'm still doing God, I can't stand it. Yeah, I wasn't sure if you wanted me to interrupt or save them to the end.
If there are questions, we can save them to the end. I just in case I was completely off base with the things I was talking about.
No, it's one question came in. Nobody is revolting against your presentation.
Okay,
yet.
Good. No revolting. Excellent. Well, let me tell you about a couple of the projects that I worked on when I was an undergrad. So again, University of Minnesota. So, there's like farm country there. And I was specifically in a lab that was in a veterinary school. So it was particularly a veterinary pathobiology lab. So studying diseases of animals, particularly large animals. So what we looked at is what were the causes of specific diseases. So I'm going to tell you about those diseases on the next slide. But then what we did is we would develop genetic tests to look for animals that could be susceptible or have the disease. And what was super cool about this was that I was taking the information and I was applying it. And I was talking to, say, breeders of animals, particularly horses and sheep, and I was telling them the research results, and it mattered. It actually was making a difference in how they were breeding their animals and the future of some of their farms. So it was really cool to see science in action in a way that it mattered. So some of my projects were, I studied porcine stress syndrome, which is a model of when you go into surgery and you have anesthesia. Some people actually have a mutation in their muscles that keeps their muscles contracted and their temperature spikes up and they can actually die on the table. So it turns out that pigs have this same mutation. And when they get stressed, like when they're moved from the farms to, say, a slaughterhouse, they also go through this reaction, and apparently it destroys the meat when they seize up. So it was nice to be able, we could use the pigs to study this human disease and also something important for pig health. So I studied pigs. I also studied a disease in sheep called scrapie. You might know it better in cows as mad cow disease. And the third one that I spent most of my time on is a disease you've never heard on unless you raise paint horses, which I didn't know what a paint horse was. Think of a bucket of paint, brown horse, throw the white paint on it. That's a paint horse, not literally, but you get what I'm saying. This disease is actually pretty catastrophic. So it takes a long time to grow a foal. This foal comes out and it's all white. It's not albino. It still has normal colored eyes, but everything else is white. So that means it's missing the pigment cells. These are called melanocytes. So the problem in this syndrome is that the neurons that are supposed to innervate all of your body, they go all the way through the small intestine, but they don't quite get to the large intestine. So what happens when these foals are born is that they suckle. So they're taking in food from their mother, and the food goes into the stomach, yay, goes in the small intestine, yay, and then it doesn't get into the large intestine. And they start to get impacted, and they can die within 24 to say 72 hours. And it's really, really sad. And it turns out that it's a mutation, it's a recessive mutation that we found and then developed a genetic test to. So I could talk to the breeders on the phone and say, yeah, don't mate those two animals together because you'd have X% chance of creating one of these foals. Instead, mate these two animals together and you will totally avoid this disease. So here's a picture of me at my first national meeting. We got to go to San Diego, which I was like, wait, I get to go to a different place that's warm in the winter, not Minnesota, and I get to talk about my research and you pay me for it? What? Yeah. So what I learned through this experience is that, man, I really loved research. It was much better than book learning. Sorry. And I was pretty good at it. I really liked it. I could disappear into an experiment. I could do multiple experiments at the same time, and it was so calming. And I learned about really the research, the impact of research on the community. And this was really important to me because there seemed to be a disconnect in what researchers were doing and how it actually benefited society. And that science really does need people who can engage and communicate with students, because some of my classes that I took, woof, so boring. And at the end of my undergrad, I was like, yeah, large animals, no thanks. I don't want to work on those anymore, because you can imagine that many of the animals had to be put down in our experiments, and that I just didn't think that I could carry that with me anymore. So in addition to my idea of a scientist, I started adding words that kind of, when I grow up, or things that define me. So I'm going to kind of build on this picture to kind of show you where I am at the end. So at the end of undergrad, I had some decisions to make. So I could keep working in the same lab. In fact, Mick tried to tell me to just stay here and just do a PhD. You're already way ahead of everybody. You could get a PhD in a really short amount of time. I was also thinking about maybe getting an MD, because I wanted to do research with people, and I thought maybe a background in MD would help my knowledge base and also help the types of experiments that I wanted to do. I was dating a dude for all the way through undergrad, and he was a material scientist, big smarty pants, and he was going to go to graduate school in Illinois. So I could follow him there and maybe just go to graduate school there, or maybe I could take a year off. And since I had like powered all the way through undergrad and I was working the whole time in the lab, I felt like I was a little bit frazzled and I needed a break. So I decided not to keep working in the lab. I didn't follow the boyfriend. I didn't want to get married at the time. I wasn't ready for that. So I said, bye-bye. I didn't get to go for an MD because it turns out I don't really like to touch people that much. And that's kind of a big part of some of the stuff you got to do when you get an MD. So I thought, no, if it's just the knowledge, I can probably supplement that information elsewhere. And I thought maybe I'd get a PhD, but I needed some time off. So I actually moved out to Boulder, Colorado, because I had a friend from high school that lived out there. I didn't know anybody else. I moved in with 4 guys that I didn't know. They had a trampoline in the back. That was fun. And I ended up working as a bartender and a janitor. So a bartender near Boulder and then a janitor up in the mountains so that I could get a free ski pass. And I just lived for a year and it was lovely. I will say about 6 months into it, I realized that my brain was kind of starving for, a little bit more engagement. I used to memorize all the songs on the jukebox and I knew their four number numerical code. Not a good sign. So I started to apply to graduate school and I looked all over the country. I interviewed at a lot of places and I ultimately decided to stay in Boulder because I don't know if you've ever been there, but it is really beautiful. And I really like the vibe. Everybody was about the environment. Everybody's out about, being healthy. And I felt like I belonged. And when the snow came, the snow was fun versus when the snow came in Minnesota, the snow was not. So it took me about 5.5 years to get my PhD. I was the first in my class to graduate, even though I failed my oral exam at the end of my second year. And I really thought about leaving my PhD at that time. And the focus was I studied DNA damage checkpoints and how cells go through cell division. So I switched to the fruit fly model. So on the bottom left there is a male fruit fly, Drosophila melanogaster. And this is a great model for looking at cell biology and how cells interact and communicate with each other. So the process that I studied is the one here. So just one diploid cell, you duplicate the chromosomes and then the chromosomes divide and hopefully you get clones of the parent cell. So 2 daughter cells. And what I studied was what happens to this process when they're damaged, say in the early part of the cell cycle or the later part when the chromosomes are actually trying to separate. And why this is important, particularly to human health, is that the processes that control this pathway generally are mutated in cancer. So we're able to kind of look at those high level questions just in an organism that has fewer genes. It's a little bit simpler than humans. So what I really liked about this time is that I got to look at genes that were important for human health in a different system, and then kind of look at what the consequence of if these genes aren't working properly. So what I learned during graduate school is that microscopes are awesome. Microscopy generally is awesome. So I like to watch mitosis through a microscope. Great. I also got to do a lot of teaching and mentorship during graduate school, and I found that that was as or more rewarding than what I was doing with my hands. And I really liked research, but I was starting to think that there might be something more out there for me, but I wasn't exactly sure what that was. And although I made the move into fruit flies because I wanted to get rid of the large animals, I was kind of annoyed that the fruit flies weren't deemed as medically relevant as, say, working on a mammal. So I kind of made a switch again. So here I'm just adding a couple other words, like now I define myself as an educator and a cell biologist and all this other fun stuff. So at the end of graduate school, kind of at a crossroads again. As I told you, when you get a PhD, you ain't done. You have to go get this thing called a postdoc most of the time, depending on what you want to do. So I was like, well, should I keep working on cell division? It's kind of cool, but I want to expand a little bit more. Should I just go and get a job and say at a company and start making lots of money? My friends did that. They still make more money than me, whatever. I had a friend that also failed his oral exam in graduate school and he decided to move to Tanzania to be in the Peace Corps. Amazing. I ended up not doing that because it's Tanzania and that was really far away. And then I thought maybe I could just get a job teaching. I wasn't quite sure. So I interviewed a lot of people. I asked them, kind of what their jobs are like. And ultimately, I decided not to keep working in the same field. I didn't just go get a job. I decided to go and get a postdoc, definitely not join the Peace Corps. So for graduate school, I had actually interviewed at University of Pennsylvania and decided, who would want to live there? So, I stayed in Colorado. And it turns out the job I found for my postdoc was actually at Fox Chase Cancer Center, which was in where? Philadelphia. And it was only going to be 3 years or something. And by the way, that was 2005. And if you do the math, that was definitely more than 3 years ago. But anyway, so I came to Fox Chase Cancer Center, which is in Northeast Philadelphia. So it's a comprehensive cancer center. So they treat patients Only adults, no kids, so you have to be 18 years old to be treated here. We treat virtually every type of cancer. We're a research center and we're a training center. So what I studied during my postdoc is kind of shown here. So these are still images from a movie that I made. And the little green guy is the natural killer cell. So again, this white blood cell goes around your body looking for stuff to kill, hence the name. And then the red guy is a tumor cell. And what you're looking at here is the NK cell kind of getting close to the tumor cell, touching the cell surface, going, are you the right one? Are you the right one? And then it gets real close to it and gives it a hug and again tells it to kill itself. And what you don't see, because I cut those images out, is that the tumor cell then goes through cell death. And again, this was fascinating. This happens in minutes and it was so cool. You could see this. And the NK cells will then come off the tumor cells and they can kill a few other tumor cells. And then sometimes the NK cell gets exhausted and it dies. It was amazing to watch this. And I could do this all day. As you can see, hopefully you see that I'm excited. But I was studying human cells and I was really trying to look at how cells communicated and how to get the NK cells more active. Because I don't know if you've heard of CAR T cells, C-A-R T cells, kind of like the car that you drive. So NK cells, like CAR T cells, can be modulated and be used in cancer patients so the patients can fight their own cancer. Fascinating. Anyway, so what I learned during my postdoc is that research is cool. I don't want to do it. I don't want that to be the only thing that I do. I don't want to go and be a research faculty member because I felt like I either wasn't good enough or it would take too much of me that I couldn't do anything else. So there turned out to be a lot that I could do with my skills. And I knew that I was a pretty good speaker and I really liked to mentor. And I wanted to do something where I could be a mentor, a teacher, and a researcher, maybe at the same time. But I didn't think my skills in teaching were as top-notch as they needed to be. So I took a little time and I started working at Monco in Bluebell. And I taught for a couple years in intro to biology class for non-majors. And I loved it. I loved it as much as the lab. Just seeing when people understand something for the first time and you're like, yeah, that was awesome. And I felt so lucky to find yet another thing that would get me up in the morning. So I thought, all right, there's got to be a way that I can kind of put these things together. So this is kind of how I think about myself now. So I'm a scientist at my core, but I do all of these other things and they're really driven by what either I'm curious about, that I struggle with, or I want to learn more about, or that get thrown into my lap. Okay, so that's kind of the end of part one. So that's a little bit about me. So let me tell you about, that is not the end of part one, I lied. That's near the end of part one. So let me tell you a little bit about my current job. So I'm the director of academic affairs. What that means is I direct a whole department. And in that department, we have lots of programs. So I train high school students, undergrads, and then all the way up to postgraduate on the research and the medical side. So my favorite thing, unless my undergrads are listening, then their thing is my favorite, but is my high school program. So the Teen Research Internship program. It's so much fun. I love learning from the students as much as they're learning from me. And it's just great to mess around in the lab. So here are just some pictures of various programs that I run. I also, in addition to running programs and teaching, I do a lot of community outreach. So for instance, in the summer, I run this Careers in Science and Medicine. If you're interested in learning about more careers that could be at cancer centers such as Fox Chase, tune in for that. Last year, I held them all through Zoom and then I archived them so they're freely available on Fox Chase's website, but they're buried. You'd never find them. So I'll put the link into the chat box later if somebody reminds me. Somebody out there, remind me. All right, I also do a lot of writing and a lot of editing, and I'd say that my writing skills were not that great kind of at the beginning. Like science writing was okay, but like writing an email, I write those all the time. And you can ask any of my students. Sometimes I don't respond to their emails in time, but whatever. I apologize. So I write newsletters, lots of publications, and social media. Oh man, yes. I have to apply for grants a lot. I have to give presentations kind of like this. I sometimes give tours of the Cancer Center. I serve on committees like Dave and I were talking about so that we can connect with other educators and help them with their programs. And I do bench work, but not as much, hardly ever. It's rare. So I kind of put these into two buckets of what I do is I manage and mentor other people. And I think about the future and building and problem solving. So what I love about my current job is that I'm constantly growing and changing and learning new things. I get to mentor people and kind of see the development of other people. I get to think about science, but I don't have to necessarily do the science, which is nice. And I feel like the work that I'm doing now versus the work that I was doing at the bench actually makes a difference. So kind of that thing that I told you at the beginning about telling the breeders about the genetic results from their animals and that it was informing them to make a decision. I got that feeling back of what I'm doing right now. So this job is also great because it's a challenge. And I'm constantly learning and growing. On the flip side, like I said, always answering emails, and I can't keep on top of it. It irritates me. They're always there. And the more I answer them, the more there are. And I just feel constantly bad about it. I also can't help everyone. Like for instance, for my summer program for this summer, we had only a few spots open because we had to grandfather people in from last year, and we had like 125 people apply for very few spots, and all of those people are excited, and you have to say no to a lot of people, and I don't like that. And also, since I'm constantly trying new things, I'm in uncharted territory. So I'm plagued by self-doubt. I don't know if you've heard of imposter syndrome, but I got that thing in spades. So a lot of times I'm like, oh, well, I don't know how to be the director of a department. I never got a degree in that. I never got training in that, but I'm doing it. So fake it till you make it. I do really miss being able to disappear into an experiment because that was really easy. And my job on a day-to-day basis can be really hard and challenging because a lot of times money is really limited and I can't do all of the things that I want. So what skills do you need to do my type of job? So any of the jobs that I told you about, graduate school, a graduate student, postdoc, teacher, director. So you really need to be an excellent communicator, not just written, not just orally, but also graphically as well. So through images. And you also need to be able to engage different people. So I talk to owners of companies. I talk to students, I talk to peers, and I have to figure out how to engage each level. I also have to multitask, and sometimes I don't do that very well. And I have to always be solving problems. And the last thing I think here is true. I have to be able to, you have to, if you want to do this type of job, embrace failure and learn from it. Be like, it's okay not to be good at anything all the time. And sometimes that's hard. That is not a comfortable place for most people to be. All right, so how do you get here? Well, explore, try stuff out, do internships, volunteer. You might not like it. And don't just do things that you think you're already gonna like. Try things that you think you might not like. Keep a list of what you like versus what you don't like and keep revisiting it. And if you like lab work, seriously, go ask somebody to volunteer in their lab. Tell them how great their research is. People love to talk about themselves and love to hear about their research. So let me tell you just a little bit about TRIP. So I want to take maybe 10 more minutes and then we'll leave it up for questions. So I'm going to tell you what TRIP is, how you can get involved, and give you some ideas of some of the experiments. So TRIP, really, we created it because the goal here is to foster enthusiasm for hands-on learning in the lab and foster enthusiasm for science. So in this program, each student develops their own project. So it's completely open inquiry. And what I love about that is people get to be curious and they do something that matters to them. That's really key. And throughout, even though it's a research course, we're really promoting science communication through presentations and blogging. And also we explore different careers. So I'm trying to do like one-stop shopping with the students that are in the TRIP initiative to give them sort of a survey of what research and science is about. And importantly, these students are hanging out with other ones that are really excited about the same things. And so you get to build a community of like-minded people. So some people that I've met in Trip have now gone on to college and room together. So I think the relationships that are built are big and true and unique. So importantly, the program is free. Even though it costs Fox Chase a lot of money to offer this program, it's key that it's free for the community so that anybody, back to that idea about diversity, that anybody can be involved in this program. So I run it twice a year. I run it now on Saturdays at William Tennant High School. That's this thing here in Warminster, and also in the summer at Temple University. So in each session, there's 8 students, and I usually have myself and another instructor who's a PhD scientist. Sometimes I have graduate students, and sometimes I have undergrads. And then I usually have alumni that have been involved in the course come back as teaching assistants. And there's about 10 or more sessions for each course, and each session is about 4 hours. So this one runs from January to April, and this one runs July. So to get into Trip, you have to complete an online application. It's essay-based, and you have to submit a number of letters of recommendations. We're kind of tweaking these numbers. And then your applications are reviewed, and people who qualify then have to interview. So it's kind of a long process, and it's just basically getting to know people. Do I want to spend my Saturdays with you? So if you want to find out more about Trip, here's the website. So it's tripinitiative.com. On our website, you can see a gallery of the alumni. I apologize, it's a little out of date because, I've been answering emails. Although we just closed the application cycle on Sunday, the next one will open like in September, but you can kind of tool around on the website and look at the blogs that the students write. So what do you do in Trip? So again, Trip, by the way, stands for that. Oh, sorry. So Trip is all of these things, like I said. What we do in Trip is we use this little guy, fruit fly, and it's a female on the left and a male on the right. You can tell the male because they got a big old black butt. So what we do is we mess with the flies. We change their environment, we stress them, we change their diet, basically anything that the students can think of that's not illegal, and we can do it for not too much money. And then we look at what happens to, say, development. So how the flies develop, how their cell division and cell differentiation is affected. And then we can also look at their behavior. So how do they move? How do they respond to their environment or sense their environment? How do they learn? All of these things, really, really cool. So far we've trained over 160 students, and we just added 17 more. I should have corrected that. And again, these students come from all over the region, actually. So really the goal of Trip is to have every student be able to ask and develop their own question, and then talk about their results. So every student gives a talk in a symposium at the end of the program to their peers, to their family, and to some people from Fox Chase. In addition, they learn how to communicate. So I'm flipping through these kind of fast, but in addition to communicating orally, they also create graphics and other versions of communication and put those out into the space. So for instance, we have a blog section on our website that you can learn about the program from the student's perspective. You can listen to me all day long. I wouldn't recommend it, but you can go look about what the program is about directly from the students. So some of the blogs look like this. So this was Abby Shen's project, and she was looking at the role of caffeine when flies aren't allowed to sleep and what happens to their anxiety levels. So again, students get to develop their own product. They get to think critically, and they importantly get to fail in a safe environment and learn how to think through it. So kind of building skills that they need for pretty much anything else that they want to go into. And again, I'm just going to skip ahead here. Is just what's important about Trip is that students get to apply, what they're learning about biology to their projects and challenge themselves. So another thing that's fun for me is that I get to teach people how to teach Trip. So I've got to work with other educators like Mr. Ignacio Ohio, who's at Tennant, And this is Bob Herbstrich from Central, but also I get to work with researchers and people that want to be teachers. So for interest, Miss Trinity Pellegrin is now teaching her second year of high school. And some of these ladies and a couple of these dudes are research scientists that also want to be interested in teaching. Anyway, so let me just tell you a couple things about what we can do in the lab. So if we have students that are interested in study development, say they answer, yes, I'm interested in studying development. Well, the next question I ask is which phase? So just here's fly development over here. So you got a female fly, you got a male fly, they hold hands, they dance, they do something, and you get an embryo. So after about 24 hours, this embryo hatches into larva. You may call it a maggot. I call it a larva. And it develops through these successive moltings called instars, first, second, and third instar. After this last molting, going from what I call a tube with a mouth and a butt, this is the juvenile stage. So I just called all of you tubes with mouth and butt, but whatever. Then it pupates. So it goes into like a cocoon, and it goes from this tube with a mouth and a butt stage to something that actually has a head and a body and legs and wings. So we call this metamorphosis. So depending on what the student is really excited about, I'm like, well, do you want to study early, say, gonad development? Or the egg or sperm development? Do you want to study embryos? Do you want to study pupa? And we have assays that are built into the program, either established or the students develop them themselves to kind of ask interesting questions. Similarly, if students are interested in behavior, I ask, well, what kind? Are you interested in how animals sense their environment? So they see, they smell, they touch, any of that? Are you interested in how flies react in terms of their mood? So flies are happy, they can get anxiety. They can also get things like addicted to alcohol. So they can, flies is a great model system for studying addiction. Or maybe you're interested in how flies, move. So again, I'm just cherry picking a couple assays here and just showing you the names, not that important, just to show you that we have so many ways that we can ask these types of questions. So let me just tell you an example of just one project here, and then I'll shut up. So I want to tell you about Ifena. So Ifena was a rising sophomore at Cheltenham High School. He now is graduating this year. And he was really excited to look at fertility. So his mom actually struggled in conceiving him. And when she was trying to conceive him, she took 2 over-the-counter substances called maca and Vitex. I had never heard of these things before, so he brought them into the lab. And he said, Dr. Purdy, I want to know what happens to female fertility when I feed these things to the fly. I'm like, sweet, let's give this a try. Nobody knows what's going to happen. Let's figure this out. So if we look at kind of Ifena's thought process here, is he wanted to look at early development. And so he used these various assays to collect data. And how we actually collect the data, again, here's the fly development, is we take embryos. So if we want to look at female fertility, we look at the number of embryos that are produced over time. And then if we want to look at later development, you can also count the number of pupa that come from the embryos. So here's just a little bit of Ifena's data. So we're looking at the number of embryos that were produced in a female under these different conditions. So if we look at the control, it was a little less than about 1/2 of an embryo per female. But you can see that at increasing concentrations of the maca, it seems that there was an increase in fertility versus with the Vitex, maybe not so much, maybe a little bit worse. But what's interesting, if you do the combo, put both of those drugs together, fertility, not so good. So I'm going to just stop there for a second and speed through a bunch of other slides that apparently I wanted to get through, way too many, and just say that if you're interested in learning more about TRIP, damn it, here are all of our handles. Again, we're on Instagram, we're on Twitter, and we're on Facebook for your parents. And thank you for your attention. If you have questions, I'd be happy to take them. And here again is my information. So here is my email address. It's just amanda.purdy@fccc.edu. So all of our email addresses at Fox Chase are the same, fccc.edu. All right, I's done.
Thanks, Amanda. Panel, so I can move on. And I would love for you to actually use your own voice and ask Dr. Purdy some questions. But let me take the ones that were in the chat box here. First one from Ankita. Sorry if I said your name wrong. How did Dr. Purdy go about getting into research labs and finding people who were willing to work with you? Great question.
Pestering, asking, calling. I actually, I visited a lab one time. I got into the building and I just walked into the person's lab and I'm like, hey, what are you guys doing? You interested in taking an undergraduate at all? I can do anything. So you would be amazed at how charmed people get if you just reach out to them and show an interest. So on Fox Chase's website, for instance, their email addresses of the faculty members are there. If you do a little bit of, Google searching, you can figure it out. So I would say reach out. And if you're in college, you can go to your counselor and say, how do I get into a lab?
And I would just add something, Amanda, just don't reach out, put in that little bit of extra effort and do a little bit of homework. So when you reach out, you sound really interested and really educated about what you're talking about. And then just another quick anecdotal story. I hired a research lab technician in one of the labs that I used to work in. She was right out of her undergraduate studies. And literally what she did, she showed up one day. She really wanted to work in this lab. It was not my lab, it was somebody else's lab, and I was also working in it. But she wanted to work there, and she came and just like spent the whole summer volunteering. But literally walked in and was like, "I want to work in this lab. What can I do?" Yep. And we ended up after the summer, we ended up officially interviewing her and gave her a job. So, great advice.
What you can't teach is excitement and interest in something. I can teach you how to pipette. I can teach you how to run an experiment. But if you don't have that fire or that passion to start, it's just not teachable. So when you approach somebody, like Dave says, be knowledgeable about what they're working on, but also show your passion.
Yeah. Mrs. Eckstein just wanted to ask about the age groups of the students in TRIP.
14 to 18 is usually the range. The sweet spot is kind of around, sophomore, junior year. It's helpful if they have had some math or some chemistry. It's not required. It's just a little bit easier for them if you have. So it's usually for high school students, but I take rising freshmen and graduating seniors as well. Great.
I think this is from another student. Sharon asks, what were the high school classes that benefited you most when studying for your bachelor's and your PhD?
Well, that's a really interesting question because it's not as straightforward, I think, as you think. So obviously I went into a science major. So those core science classes like biology and even math helped with being comfortable when I got to college. But the thing that actually helped me the most was the ones that I struggled with because I developed better study practices because of them. So it's sort of a mixed bag. So I would say if things are too easy for you, challenge yourself, take something harder so that feeling is not new to you when you get to college.
And I'm also just going to step in and give a piece of advice from my own experience. While those science classes are tremendously important and they will be required for certain majors, I like to tell students to also take classes, especially at the end of your college journey, that maybe interest you. I took 3 years of photography at the end. The first one was simply to fill my fine arts requirement, but I actually love doing photography. So I took 2 more years of it. I took a half a semester, Amanda, do not laugh and do not let Ignacio know this. I took a half a semester of jazz tap class because I'd never danced before and I wanted to see what tapping was like. So that's
awesome.
I could have taken analytical chemistry if I wanted to, but I chose to take photography and tap class because that was why I was in college too, is to get some other experiences. So science classes, they're going to be important. You're going to need to take a bunch of them, but don't let that overtake your desire and passion to try something else too.
And also English and writing. I use writing every step of my career. I can be better at it. So I would take those classes. Take more of those classes than you need.
Well, that leads into our next question. How did you develop good communication skills?
Practice. There's no other way to do it. And you're not good at it at first.
Oh,
No. My first, my first talk that I gave in graduate school, I was supposed to give a 10-minute talk, and I was giving the practice to my lab, and it was like 3 minutes, and it was missing all the important information. And I was like, and we're done. And they're like, yeah, so you didn't actually start us anywhere. You didn't give us an expectation, and you didn't tell us what we needed to know. So all of those things come through practice. So to be a better speaker, I signed up to give talks. Was it comfortable? No. Did I like every moment of it? No. Did I get better? Yes. And I'm still tweaking. Like, have I given this type of talk before? Mm-hmm. Did I keep my slides exactly the same from last time? No. I'm always struggling to get better. So I would say, how do you get better at that stuff? Only by experience and trying, which is a crappy answer. I apologize, but it's the truth.
And you're here today on a Friday at 3 o'clock in the afternoon when it's 75 degrees outside. So I can imagine a lot of you on this call today are very great students, very motivated, right? But you might also have a very type A personality where you don't like to be wrong, and you don't like to fail, and you don't like to do things that you don't seem were putting your best foot forward. But guess what? You're going to have to do those things. Get comfortable with it. Especially in science, you're bound to fail. So get some thick skin.
Yep. Science, 90% of the time, experiments fail.
Yep.
And that is not an overestimation.
It's underestimating probably, but let's not go there.
Yes.
Thanks.
Positive.
Prima asked, what was your favorite lab you worked at?
The first one, my undergraduate lab, because everything was so new. And honestly, I didn't know what the heck I was doing, which was weird. But I like was in charge of things and got to like the conversations with the breeders. Like I felt like I knew stuff, and I was making such a difference. And I was getting to learn all of these techniques that I was learning about in class. It was just so neat to be able to do it with my hands. So that, rush of excitement that you have at first, that was my favorite. I would say the natural killer cell experiments were also pretty cool. It's just fascinating to watch cells kill each other, but that's just me.
Amanda, I do want to be respectful of your time. It's 2:59. I have some more questions. Are you willing to stay on, or do you need to go to something else?
I'm good. I don't have another meeting for, let's see. So I have to interview a postdoc in about 45 minutes.
Okay. Well, hopefully we won't be on that long.
No,
I will let you type out some emails, but students, obviously you're welcome to stay on as well. But if you have something else to do, obviously just drop off. How did you deal with being unsure of what you wanted to do?
Really good question. I don't know how to answer that one. So, and to say, just do it, sucks. So what I do when I encounter a terrible or a stressful situation is I think, what's the worst that could happen? And then I'm like, okay, I get my mind around that. And then I think, what's the best possible outcome here? And then realize it's going to be somewhere in the middle. That helps me like decrease my anxiety and think, okay, even if that horrible thing happens, I have a plan to go for that. I try to put myself in uncomfortable situations because I need to practice that. I would say that would be my answer. Again, I'm sorry if that's not like, the magic bullet, but that's how I do it.
Mm-hmm. Before I get to the next question, I do see a couple hands being raised, and I will get to those as well. Rachel, you did ask, could you please post the link for the career volunteer program? Could you just let me know in the chat or pop on, what are you talking about with the career volunteer program? I want to make sure we're giving you the right link there.
The careers in science program that you
Oh, got it. Yep.
Thank you for the reminder. I'm looking right now. All right, I'm going to put this in the chat for everybody.
All right, a couple of people that would like to ask a question. All right, I'll take Diego first, because I think I saw his hand come up first, and then Emma, I got you, don't worry.
Cool. So, hello.
Hello.
I just had a couple questions. One was specific to your NK cell study. Under the microscope, can you see the MCH1 proteins or no?
No,
Are
the
those
major histocompatibility proteins,
Yeah.
no. No,
Okay.
you can make them fluorescent, or you can use like a protein, an antibody or something that is fluorescent that recognizes it, but no, you can't see them.
Okay, cool. And my other question, my big question is, what is the biggest difference between undergrad, postgrad, PhD, and post-PhD in the research, like the experience between all of them?
So graduate school and postdoc is really teaching you how to think. So undergrad, you're basically kind of told what to do. In my case, it was a little different because I was precocious and I just would do stuff anyway. But graduate school, they're teaching you how to think, how to design an experiment, and how to rigorously analyze data. In postdoc, you're supposed to not only do those things, but develop your own ideas. So come up with your own projects. And the difference between the learning how to do an experiment that's part of a project and then learning how to do an experiment and coming up with the project is very different. So those are, I would say, the major differences. Did that answer your question, Diego?
Yeah, and the difference between PhD and the postdoctorate, is there a difference there? Or is it just kind of like?
It's just more training. It's generally more training and you're more independent. It can be really dependent on the person that you're working for or with and where their funding comes from. But in general, think about it kind of as a scale of like undergrad, you're learning a little bit, graduate school, you're learning more and taking more ownership. And then postdoc, you're basically supposed to be your own boss in a way. That's close.
Sorry, I'll just add onto that. In a postdoc, I think a goal is publishing papers because that's what's going to get you a faculty position down the road, is that they want to see your publication record and that you can publish well. And that goes along with designing good projects and good experiments and thinking those things through. And I think also as a postdoc, I would suggest trying to get some mentorship experience, training, like, because that's what you're going to be doing if you get through your postdoc and get your own lab. You're going to be training undergraduates, you're going to be training graduates, you're going to be training other postdocs at that point, right? So trying to develop yourself as a mentor and train other people is something that I think you could do in your postdoc as well. All right, Emma, I don't want to forget about you. Go for it.
Hi, Dr. Purdy. I apologize if you hear any noises. There are currently people walking on the roof of the building that I'm in. I was just wondering a little bit about the killer cells specifically, because I'm currently doing some personal research on sleep, and I've actually come across the effects of not getting enough sleep on the development and like function of killer cells. And so I was just interested if you've looked at any of those, like of how different circumstances or biological processes affect the killer cells.
So yes and no. So not me personally. What I have listened to a podcast, Hidden Brain by Shankar Vedantam, had a couple interesting things about overall like immune health, and showed that so your NK cells, depending on who you are, you'll have like there'll be 4% of your white blood cells up to like 25%. And this number can fluctuate in their overall activity. So we're all different. So yours might be more active than mine just at a baseline. But what they found is across the board, like everybody, when you spend time in nature, and they say it was just like 24 hours. So not looking at pictures of nature, but actually being out increased your number of NK cells pretty rapidly, and also the overall activity of them. So if you're sleep deprived, if your diet is poor, all of those things, your NK cells don't develop as well. They won't be as active. Did that answer your question?
Yeah, that's fascinating. Thank you.
All right. It is fascinating, right? Podcasts. Those are so great. Hidden Brain. You're welcome, Shankar. You're welcome.
Final question I see in the chat box right now, unless anything else comes through, is what do you look for in high school students that want to help out in the lab besides interest?
Besides interest. Yeah. So that's tough. So, and I don't want to say that it necessarily depends on the day. I want to know that somebody's done their research so that they are taking the conversation with me seriously, that they didn't, like you said, show up and just be excited that you told people not to do. that they've read about what we do, that they understand it to a level that an undergrad or high school student would, and that they come with questions, and that they have a realistic expectation about what can be done. So what I always tell students when they're reaching out to faculty here to maybe work in their lab is that it's a big ask to work in somebody's lab. They have to train you, and you have only limited amounts of time. I don't know why I keep looking at my fingers and touching that, but you have a limited amount of time that you'll actually be in the lab. So you have to figure out a way to come across like, I am going to add value to your lab. I'm not just going to be a burden on time and resources. So I think how you can do that is by showing up and being prepared and being excited, and think about what you're going to get out of that experience and what they can get from you. Hopefully that answered the question.
Yeah, and I hate to keep adding on, but these days, in this informal science education space that Amanda and I are in, we hear a lot about life skills, employability skills, whatever you want to call them, soft skills. Those are important, and we've noticed, or I've noticed and heard that, you know, a lot of students unfortunately are starting to lack some of those soft skills. Like, be on time, put your phone down when you're in the interview. Don't check it, or don't feel like you need to snap out to somebody that you're in the lab. Like, be professional. It's going to help and go a long way as well.
Mm-hmm. Yeah, when you send me an email, hi, Dr. Purdy, comma. I attended your webinar, blah, 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 blah. So you give me a little context. I wanted to learn a little bit more than that, instead of, hey, how's it going? My name is such and such, and being like really familiar. Like, just spend some time being professional about your communications. Don't text me in an email. Like,
useful sentences.
Yes, useful
no
sentences.
Abbreviations. I don't know what IDK means. I do know what that one means, but I have two middle school daughters, and I look at their text threads and I'm like, what are they even saying to each other? But don't use that in an email to a professional. It
was a Jeopardy category the other night.
Really? That's great.
And I was like, I don't know that. I don't know that either.
Yeah, and Mrs. Eckstein said that social distancing hasn't helped people's social skills and soft skills these days either. So I just want to say that I haven't seen any more questions come in. I don't see any more hands raised. Feel free, as long as Dr. Purdy is able to stay on. I'm able to stay on as well for a while. I did put my email address in the chat box as well. This was recorded. If you wanted to go back and review anything, reach out to me. I don't know if I asked for your email addresses in the registration. If I did, I can always send it out that way. If not, you might have to reach back out to me. And I encourage you, we have at least two more or three more of these webinars. So if you'd like to learn from other science professionals that happen to be all women, it's great. And feel free to come back next week. And I can give you the schedule, which is why I said, reach out to me. That's my point.
Friday.
Friday. Diego, is that another hand raise, or is that one from before?
No, this is a new one. I just have a quick question for Dr.
It's
Purdy.
his other hand.
Oh, yeah. This is just for a personal standpoint. Where do you get your research? Like, where do you get all of your papers and stuff at like a doctoral level that like people can read? Or, like,
so where are they housed? Like, where can you download them?
Yeah, because I've been looking for studies, but they're always like under like a wall and stuff. I just want to like read.
Yeah. So our papers are published in journals, and some journals are open access, so anybody can get them, or they're not open access, and you have to have a subscription. So what's really nice is if you're associated with an institution like mine, our institution has subscriptions to a lot of things. So if there's something that you're interested in that Fox Chase has a subscription, just go ahead and reach out to me. Hi, Dr. Purdy. Remember, my name is Diego. I attended your webinar, and I was, interested in reading about this, but I don't have access. You can reach out to people and have them download the PDF and give that to you. Otherwise, there's a lot of times just open access journals in general, and more people are pushing to have things in open access. Also, to get our publications out earlier, this thing called bioRxiv.
bioRxiv.
Thank you. I was like, no, brain, shut off. And that anybody can access, correct?
Yep.
Yeah.
That's like the epitome of open access right there. And those
Wow.
aren't even peer-reviewed papers either. So, I think it's of good intention, but you do have to be aware that they haven't been through the scientific vetting process quite as rigorously as some of the other published journals are. And Diego, and everybody else, but you can go old school too. And every author has an email address associated with the author line or the
Corresponding author.
Yep. Write to them and be like, hey, I saw your paper. Can you please send me a PDF or what we used to call a preprint of the paper?
Yep.
And most people would be happy to send that to you as well.
Again, people love talking about themselves.
Oh, yes.
Love talking about their research. So if you show an interest in it, they'll be like, oh yeah, I'll give you all my papers. Want this one too?
Got it. Thank you guys so much.
You're welcome.
You're welcome. All right, I'm going to wrap this up. We've taken up enough of Dr. Purdy's time. Thank you so much, Amanda, for joining us today. That was great. I learned a lot more about you today as well. So wonderful. And thank you all for joining us on this lovely
Bye.
afternoon. Enjoy your weekends.
Thank you.
Thank you.
Thank you.
Thank you.
Have fun on your interview. Amanda, your
All right.
postdoc interview.
Yes. See you guys later. Thanks again for
All right.
the invite. And I guess we'll talk soon about
I'm sure.
your other stuff. All right.
Yeah.
Bye.
See ya.